next presentation, which is from Scovia Adikini in Uganda. Thank you.
Good morning. I'm called Scovia Adikini from National Arid Resource Research Institute, one of the institute under National Agriculture Research Organization based in Uganda, and I'm the co-I for East African Center of Innovation for Finger Millet and Sorghum, being hosted in Uganda and implemented in partnership with Tanzania Agriculture Research Institute and Kenya Agriculture Livestock Research Organization. Today I'm presenting on harnessing sorghum and finger millet genetic resources for increased productivity and utilization in the arid and semi-arid regions of East Africa. East African semi-arid and arid region, food security and food and nutritional insecurity characterize the region, and this coupled with poverty and scarce water resources, land degradation, and gender inequality. As a consequence, over 36% of children are malnourished, and over 45% of death among children below 5 is as a result of malnutrition. Finger millet and sorghum are the predominant crop, well adapted to the region. However, the current productivity is very low. The yield is up to 1 ton per hectare compared to 3 tons per hectare, which is obtained on station. And the low yield is as a result of several factors, including dependency on rain-fed agriculture, which is characterized by low productivity, frequent drought and and sometimes devastating floods, pest and disease outbreaks, and use of inferior varieties. The region is also poorly developed in terms of input and output markets, and also has limited infrastructure, as well as limited access to financial markets and opportunity for commercial growth. In order to address such challenges, strategic interventions and actions that are efficient and cost-effective are needed to address these challenges amidst the climate effects. And East African Center of Innovation for Finger Millet and Sorghum will provide a framework for national breeding programs to leverage resources, strengthen synergies, and harness opportunities to accelerate development, release, promotion, and adoption of varieties that address the needs of women and men in the region. The center will do this by bringing together the best tools, technologies, methods, and expertise to empower communities residing in some arid region to transition from a subsistence existence to one of increasing incomes, resiliency, and food and nutritional security. By doing so, the center will be addressing both the Innovation Lab for Crop Improvement goal as well as Sustainable Development goal. The scope of the Center for Innovation for Finger Millet and Sorghum will achieve its goal by prioritizing resources through product profile-based breeding, by conducting value chains and market analysis to identify variety characteristics and traits preferenced by men and women. It will utilize the existing genetic diversity of finger millet and sorghum in the region for the discovery of novel and important traits. The center will also focus on genotyping, developing, and using molecular marker for identification and integration of novel recessive alleles into breeding lines, and will develop protocols for phenotyping and methods for precision phenotyping. The center will also focus on establishing efficient data management system using breeding management system. The objective of the center will focus on understanding the finger millet and sorghum value chains, market analysis and drivers of adoption and gender, identification of sources of resistance to biotic and abiotic stresses, and sources for nutritional quality, gene and molecular marker discovery for novel traits. We'll also focus on development and optimization of tools and methods for efficient phenotyping and rapid generation advancement of desirable traits. Establishment of digital data capture and integrated management of data is another focus of the center. Reflecting on the co-creation process, the co-creation process was based on 5 phases, which include among this priority setting. And during priority setting, we discover that the current breeding activities, most of the varieties that have been so far released under the previous effort have not had impact due to limited adoption, especially in the communities living in the semi-arid region. And this limited adoption is partly due to lack of attention to gender-responsive trait, as well as limited knowledge on the value chain and market structure of both sorghum and finger millet. Therefore, the center will ensure that sorghum and finger millet breeding is guided by a specific product profile and ensure that traits that meet the needs of stakeholders market are identified and incorporated in the ongoing breeding line so that the variety released meets the traits that are required by the different consumers. And the trait discovery phase, East Africa is the center for diversity for finger millet and sorghum. Amidst the richness of genetic diversity, most of these germplasm have not been utilized because they have not been characterized. And therefore, novel traits has not been discovered. And this, therefore, will be the focus of the center on wider germplasm evaluation for trait of interest in order to discover the novel traits, especially in terms of disease resistance and micronutrient traits. During the genomic phase of co-creation, currently finger millet and sorghum improvement programs solely based on conventional breeding. And this sometimes require a lot of time and lacks precision, especially for recessive trait. Use of molecular tools such as marker-assisted selection and genomic selection has been successfully applied in cereal crops, but this has not been the case in finger millet and sorghum. This has not been utilized. The marker-assisted selection and genomic selection has not been utilized due to lack of reliable phenotyping data and the limited number of molecular markers for traits of interest, as well as lack of infrastructure and human capacity to carry out molecular breeding. And therefore, the center will focus on genotyping the existing breeding lines and potential germplasm so as to develop markers and develop breeding population to be used in marker-assisted selection and genomic selection. And during phenomic phase, most of the phenotyping facilities are poor in terms of and cannot generate precision data. For example, most East African, most of the breeding programs lack necessary facilities. For example, necessary screening for biotic and abiotic stress is weather dependent, and yet establishing of irrigation system would increase the number of necessary screening and reduce the screening cycle, which is key for increasing genetic gain. Post-harvest data collection is another bottleneck as it delays the process of decision making due to lack of threshing and cleaning equipment and also the seed counter. Automating this process would reduce drudgery and improve the efficiency and accuracy of data collection by reducing errors due to mixing, due to human error during the operation. And during the breeding informatic co-creation, the breeding informatics requires integration of both phenotypic and genomic data through simulation and prediction. in order for rapid and effective decision making. However, this requires efficient data management system, and therefore breeding management system will be deployed, and this will allow for the storage and management of pedigrees and phenotypic data dynamically and in full synchronization with the breeding steps. The work plan for this year is based on, I've grouped it into different objectives, and objective 1, we shall consider value chain mapping for both finger millet and sorghum, trait preference, individual survey, in-depth gender and social difference study, literature review through online search and key format interview. We shall conduct demand study survey. And under objective 2, we shall assemble breeding materials across the region, screening of finger millet, germplasm material for blast disease and national infection. We shall also do screening of finger millet germplasm for striga weed tolerance, screening of sorghum material for leaf blast and anthracnose, screening of sorghum material for striga tolerance, micronutrient analysis of the released and advanced breeding material for finger millet and sorghum. We shall also introduce nutrient-dense advanced lines of sorghum and finger millet from ILCI and evaluate them under different locations for adaptability and acceptability. Under objective 3, we shall prepare and send finger millet and sorghum for genotyping at Innovation Lab for crop improvement, establish the genetic diversity of finger millet and sorghum germplasm, develop marker panel for pedigree verification, establish crossing block to generate biparental population, test for hybridity using morphological and molecular markers, and training of technicians and breeder in molecular analysis. And objective 4, we shall procure and install irrigation facility, procure and install equipment for service handling and data capture, procure and install breeding management system, cloud server, and data collection equipment, as well as assorted lab equipment and consumable. And objective 5, we shall review infrastructure for each partner and establish an upgrade plan as required, conduct inventory and quality assessment for historical data, and migration of selected historical data for each program as required. We shall review breeding nomenclature as well as crop ontology with partners to standardize and harmonize breeding practice as needed. And we shall establish breeding management system instance for each partner. And we shall conduct online support after the cropping cycle by the integrated breeding platform to ensure correct data capture strategies and upload in the BMS. We shall participate in breeding management system training and training of technical staff for each partner to support basic IT. Under project management, we shall recruit project staff, recruitment of graduate students, develop monitoring, evaluation, and learning tool, conduct inception meeting, finalize contract agreement with Innovation Lab for crop improvement, finalize sub-hours contract agreement, and conduct quarterly meetings and annual review meetings. Immediate needs from Innovation Lab for crop improvement as a Center for Innovation for Finger Millet and Sorghum team. We shall need to finalize support to finalize the contract agreement and ensure immediate fund release, develop robust tools and methods for value chain and market analysis, develop robust tool and method for conducting gender study, develop tools and method for higher throughput phenotyping for drought, striga and diseases, standardize experimental design, data collection and management for trait discovery, and guide on genotyping processes. The expected progress, we expect to have value chain actors and market for sorghum and finger millet characterized, gender segregated quality trait preference identified and documented, gender and social differences for sorghum and finger millet crops and product documented, demand chain for sorghum and finger millet products and knowledge gap documented, genotypes of finger millet and sorghum that are candidate for resistance to pests, identified and nutrient-dense lines of sorghum and finger millet identified. Data for Finger millet and sorghum diversity generated. We should also expect to have markup panel for degree verification and marker system selection, and irrigation facility installed and operationalized, post-harvest process and data management of the breeding program organized. We expect digital data collection adopted by the national breeding programs and data management system for national sorghum and finger millet breeding program in East Africa operationalized. We want to thank Feed the Future for supporting the centers, as well as the US government and partners for availing the fund to conduct this activity. We also extend our support to our implementing partners who are currently collaborating with us to see that the center is put into shape and takes its course. Thank you very much for listening.
Thank you very much, Scovia, for your presentation. At this time, I welcome questions. George Birigua?
Thank you, Vera, for allowing me. And Scovia, thank you so much for that great presentation. This question goes to Scovia and the other colleague from Senegal who presented on sorghum. Scovia, you are right by saying that one of the reasons why adoption is low is because of non-involvement of the end users. But what has not come out in your presentation and the other colleague from Senegal is how you don't involve the brewery industry. It plays an important role in sorghum uptake. We have seen this in a number of countries, here in Kenya, Uganda, even Nigeria. I'm not so sure whether it is because of the USAID policy or otherwise. That's one. Number two, again, Scovia, in your presentation, it is not very clear to me whether you plan to focus on sorghum hybrids or just OPVs. That point did come out. Request for some clarification. Thank you, and over.
Scovia?
Yeah.
You're muted.
Unmute.
You have to unmute yourself.
Okay, thank you very much. You're asking how I will involve the breweries industry. I think in our product profile, we have, well, that did not present it here, but we highlighted one of the target product profile is on the brewery industry, as well as feed and food. For the case of the brewery industry, I want to say that the breeding program in Uganda have made progress and released some of the varieties that are very key for the brewing industry. But I think one of the things that has lagged behind is the aspect of the seed system. It's not very strong, it's still weak, and that, if addressed, would actually pull the aspect of brewing industry. But as a breeding program, we already have lines that is already released that fit the brewing industry. On our focus of breeding work that we do currently, we are not so much focused on hybrid. We are still doing much of the OPV breeding, but from last year, we've started some pipeline of hybrid, but it will not be the focus currently of the center because it is a new process and we are trying to shape it up But the center currently, the current focus, we shall still focus on the OPV by looking at what we have currently and identifying those traits that is very good that we can use to improve the existing varieties. However, the hybrid part, it is also running parallel, but it will take shape over time. We got some of the hybrid line from ICRISAT, and we are working hand in hand with the ICRISAT team on that. Thank you.
Thank you, Scovia. Ed, can you go ahead and ask your question, please?
Yeah, Scovia, I was wondering, because Striga has a lot of issues in competition with what the nitrogen inputs are. I was wondering how you all are working with the differential inputs on your research farm versus smallholders and evaluating Striga resistance.
Yeah, thank you very much. For the issue of Striga, as you said, it has a lot of issues with the nitrogen, and most of our smallholder farmers' soil are actually depleted, and the Striga is a very huge challenge to the communities out there. We have currently what we are doing, we have developed methods of soil fertility management, especially use of microdose, that we are trying to put to the farmers to improve their yield. However, beyond that, we are screening our materials for the Striga resistance. Especially in sorghum, we have managed to release some varieties that are tolerant to Striga, and farmers have taken them up. But for the case of finger millet, we are still screening some of our germplasm to select for the Striga tolerance. Thank you.
Thank you.
Thank you, Scovia and Ed, for that question. Dee, can you unmute yourself and ask your question, please?
Sure. I was interested, and this follows a little bit on one of the earlier questions. I was interested in whether you could give an example of how the lack of attention to gender responsive traits that you mentioned in your presentation has led to limited adoption. I think it would just help in everyone's understanding if you could offer an example of that. Thank you.
Yeah, thank you very much. Where we have said lack of attention in gender, I can give an example. We have our community in some arid in Karamoja, and we have bred these varieties that are very high yielding, and yeah, they perform well. They are even tolerant to Striga. But when you go to them, they will tell you, no, that variety, because yeah, it is high yielding, but it will not store for long. When we grow it, it gets spoiled so easily as compared to our variety, which when we grow, even if we don't store it well, it will stay with us for some time. So I think during the development process, we did not have that kind of a view from the community that it would guide the breeding process and it would actually address their need. I hope I've addressed, give an example for such kind of thing.
Yes, that's very helpful.
Have I
Thank
answered
you.
your question?
You have. Thank you.
All right,
Yeah,
thank you both. Next, we'll pick the question from Mike. Mike, you wanna chime in or mute yourself? Ask the question.
Thank you, Sarah. You can hear me?
We can hear you okay.
Great. Thanks a lot, Sarah. So Scovia, very nice presentation. Enjoyed it. I also was able to watch a recording this past week as well. So I've had the opportunity to see your presentation twice, and it's even better this time around. So just out of curiosity, what are the factors that you think are going to be challenging to overcome as you begin to introduce high throughput phenotyping type tools into your breeding and research program?
Yeah, thank you very much. The factors that will be challenging when introducing high throughput, one of the quick one is technical, because currently what the staffs are used to may not be the same. So unless they get trained up in using some of these high throughput technology, they may not be able to deliver. It will be just another problem altogether. So capacity building issue is really very key in addressing this. And another factor that may be challenging, maybe in terms of currently there's still limited infrastructure availability. So much as some may be introduced, I can give an example. If we say we have a good irrigation system, and then we need the whole system. If we are introducing this, it has to be a linkage, a whole system from the start up to the end. But with current resources that we have, we may only start the pilot and may not fill all the gap from the initiation of breeding up to the uptake pathway. So there will be a challenge there of meeting all the requirements during the implementation. Yeah.
Great. Thank you for your answers, Scovia, and I wish you all the luck in your research and breeding progress.
Thank you. Thank you to you too, Scovia and Mike and the others that have asked questions during this session.